Today's session is on visual perception and perceptual disorders. Today's session will cover the basic principles of visual perception, how visual information is conveyed from the eye to the brain, some basic processing of visual information by the brain, and disorders of visual perception. Sensation is the detection of the simple properties of a stimulus, such as brightness, colour, warmth and sweetness, while perception is the detection of objects, their location, their movements and their background. Therefore, according to these definitions, seeing the colour red is sensation, but seeing a red apple is perception. All sensory processing follows a similar route. Information is first received by our sense organs, which normally contain receptor cells. This information, or energy from the environment, is then converted into neural activity through a process called transduction. Neural pathways then carry this information away from the receptors and towards the brain, where it is processed in a series of relay stations. It is then sent for processing in primary sensory cortex and finally to association cortex. In the case of visual processing, the chain of events looks like this. The eye contains the photoreceptors that detect areas of brightness and colours. This information is then passed along the optic nerve and is combined at the optic chiasm. It then splits according to visual field. This information is then passed to the thalamus via the optic nerve. Approximately 90% of the axons in the optic tract go to the lateral geniculate nucleus inside the thalamus. The lateral geniculate nucleus is sometimes referred to as the LGN. Information from the left visual field will go to the right LGN, and information from the right visual field will go to the left LGN. This information is then passed on to the primary visual cortex. The information flows through a cortical hierarchy within the primary visual cortex. This information is then passed on to the secondary visual cortex and then to visual association cortex, which includes the temporal and parietal lobe. Light travels in waves and photons. Photons are often referred to as particles. The wavelengths that can be seen by the human eye fall within the visible spectrum. And animals such as owls, which depend on wavelengths within the visible spectrum, cannot see in complete darkness. Okay, we need wavelengths of light which fall within the visible spectrum to be able to see. If there is no light, there is no vision. Now, wavelengths of visible light are between 380 and 760 nanometers. Okay, these wavelengths are what we can see, what we can perceive, and they are represented on the electromagnetic spectrum shown here. Now, some animals such as snakes can see infrared waves which are too long for humans to see. Wavelength and intensity are important for visual perception. Wavelength and colour are terms which are often used interchangeably, and intensity and brightness are often used interchangeably too. If we have a look at an example here, an intense light with a wavelength of 700 nanometers is referred to as a bright red light. So let's have a look at what happens with this information for us to see something. Light enters the eye. It must first pass through the cornea, which is the transparent bulge at the front of the eye. The rest of the eye is coated by a tough white membrane called the sclera. The iris consists of two bands of muscle that control the amount of light admitted into the eye. The iris gives the eye its characteristic colour, for instance brown, blue or green. The hole in the iris is called the pupil, and light passes through this hole. We can see immediately located behind the pupil is the lens. It's the bit of the pupil is the bit of lens which is visible from behind the iris. The retina contains the photoreceptors, which detects brightness and colour and covers the back of the eye. The image which we can see when it falls on the retina is upside down and reversed from left to right. It is the brain that correctly interprets this information. The ciliary muscles down here are what changes the shape of the lens to allow objects to come into focus. The brain controls the iris muscles and thus regulates the size of the pupil, constricting it in bright light and dilating it in dim light. The adjustment of pupil size in response to changes in illumination represents a compromise between sensitivity, which is the ability to detect the presence of dimly lit objects, and acuity, which is the ability to see the details of the object. When the level of illumination is high and sensitivity is thus not important, the visual system takes advantage of the situation by constricting the pupils. When the pupils are constricted, the image falling on each retina is sharper and there is a greater depth of focus. However, when the level of illumination is too low to adequately activate the receptors, the pupils dilate to let in more light, thereby sacrificing acuity and depth of focus. The shape of the cornea is fixed, but the lens is flexible. Muscles alter the shape so the eye can obtain focused images. The curvature of the cornea and lens causes images to be focused on the inner surface of the back of the eye. Now, when we direct our gaze at something near, the tension on the ligaments holding each lens in place is reduced by the contraction of the ciliary muscles, causing the lens to assume its natural cylindrical shape. This increases the ability of the lens to refract or bend light and thus bring objects into sharp focus. When we focus on a distant object, the ciliary muscles relax and the lens is flattened. Now, the process of adjusting the configuration of the lenses to bring images into focus on the retina is called accommodation. At the beginning, we mentioned that it is the photoreceptors which detect the light and that which is important for colour and brightness. They are contained within the retina, which covers the back of the eye. However, before incoming light activates the receptors, it has to travel through several layers of retinal tissue first before it reaches the photoreceptors, which distorts the incoming light. Now, this problem is somewhat minimised by the fovea. The retina is composed of 5 layers of different types of neurons or cells. Receptors consist of the cones and rods, horizontal cells, bipolar cells, amacrine cells and retinal ganglion cells. The horizontal and amacrine cells allow for communication across the receptors and bipolar cells. They help to integrate information. And by doing this, the horizontal cells are responsible for allowing the eyes to adjust to see well under both bright and dim lit conditions. Now, the rods and cones are the photoreceptors. There are approximately 130 million photoreceptors covering the retina of the eye. Rods function better than other photoreceptors in less intense light. They are sensitive to overall illumination levels and are almost entirely responsible for night vision. Cones are sensitive to colour, and there are 3 types of cones which contain different types of photopigment and respond to variation in colour in different ways. Each type of photopigment is most sensitive to light of a particular wavelength. So, for example, erythrolabe is sensitive to red light and chlorolabe is sensitive to green light. Rods and cones are involved in photon transduction, the process by which light is converted to an electrical signal. Transduction occurs when a receptor's photopigment is broken down by light of a particular wavelength. When a photon of a particular wavelength strikes a photopigment contained within the photoreceptor, the photopigment splits apart, which starts the process of transduction. This will trigger a change in the receptor's membrane potential and the receptor in question will begin communicating with the bipolar cells, which in turn communicate with the retinal ganglion cells. Now, I've mentioned the fovea a few times. The fovea is an indentation in the centre of the retina, about 1/3 of a centimetre in diameter. It is tightly packed with cones. There are no rods here. There is an almost one-to-one relationship with bipolar and ganglion cells, making it an area of maximal visual sensitivity and acuity. It is responsible for our finest, most detailed vision. When we look at a point in our visual field, we move our eyes so that the image of that point falls directly on the cone-packed fovea. Now, bipolar and ganglion cells collate information from a number of different receptors. Horizontal cells run between receptors, and amacrine cells run horizontally between bipolar and ganglion cells and help to integrate information. The axons of the retinal ganglion cells are what combine together to form the optic nerve, which sends all the visual information to the brain. Cones and rods mediate different types of vision. This is referred to as the duplexity theory of vision. Photopic vision is cone-mediated vision, and this predominates in good lighting and provides high acuity, coloured perception of the world. The output of a few cones converge on a single retinal ganglion cell. Scotopic vision is rod-mediated vision, and this predominates in dim illumination, where there is not enough light to reliably excite the cones. This type of vision lacks detail and colour, and the output of several hundred rods converge on a single retinal ganglion cell, which is why it lacks the detail. In dim light, a single ganglion cell in the rod circuit may be getting the input from many rods, whereas the same light in the cone circuit is stimulating fewer cones. The retinal ganglion cell is therefore more likely to fire in the rod circuit than the cone circuit, making the rod circuit or scotopic system very sensitive. However, the scotopic system pays for its high sensitivity with low acuity. When a retinal ganglion cell that receives input from hundreds of rods changes its firing, the brain has no way of knowing which portion of the rods contributed to the change, whereas for the photopic system, more intense light might be required to change the firing of the retinal ganglion cell. However, when it does react, there is less ambiguity about the location of the stimulus triggering the reaction. Okay, so if we take it back a step, we can see here we have the rod circuit. Now, if light were to hit these rods up here, the retinal ganglion cell might fire. But likewise, if the light were to hit these rods, the retinal ganglion cell might also fire. But the visual system would have no way of knowing where in the circuit the light was hitting. So acuity is compromised. Whereas for this cone circuit, when this cone is activated by the light, there is no doubt where on the eye this light is coming into. So acuity is much better. The distribution of cones and rods over the human retina can be seen here. Now, what's obvious from this is that there are far more rods than there are cones. The number of cones and rods per square millimetre can be seen here as a function of distance from the centre of the fovea. So if we imagine here that this is the fovea, what we can see is that there are no rods there, but there are a lot of cones there, making it an area of maximal acuity. And then we can see the distribution of rods here. Now you might notice a big gap here. This is this part of the eye. That is where the axons of the retinal ganglion cells leave the eye and form the start of the optic tract. There are no rods and cones there. This is effectively our blind spot. We can't see anything that falls within that area. The stimulus intensity is coded by the number of sensory units activated, in this case the ganglion cells which form the optic nerve, which in turn incorporates all of the receptors which innervate a particular ganglion cell. And stimulus intensity is coded by the frequency of action potentials in the ganglion cells. Now, stimulus location, as we've already seen, is coded by the pattern of stimulation across the visual receptors, and location encoding is enhanced by the influence of the horizontal and amacrine cells upon activity within receptors, bipolar and ganglion cells. As mentioned, axons from the ganglion cells form the start of the optic nerve. For this to happen, there must be a gap in the receptor layer. And this gap is what we've already said is referred to as the blind spot. It's an area at the back of the eye where there are no receptors. It is an area essentially of blindness. So to prove that you have a blind spot, you can draw two circles like this on a piece of paper. And if you hold the piece of paper up close to your face, close your left eye and stare directly at the A with your right eye, trying as hard as you can not to move your gaze. If you keep your right eye on the A and move the piece of paper a distance from your face until the black dot disappears into your blind spot, this will be about 20 centimetres away, but eventually that black dot will disappear into your blind spot. Now, I think this one is a much easier test to do. This is called completion or filling in phenomenon, and you can do something similar here. With your left eye closed, focus on the B with your right eye and move the piece of paper away. You will notice that when you do this, that the broken black line becomes whole. Okay, so that gap disappears because it disappears into your blind spot. So the visual system uses information provided by the receptors around the blind spot to fill in the gaps in your retinal image. So when the visual system detects a straight bar going into one side of the blind spot and another straight bar leaving the other side, it fills in the missing bit and you see a continuous straight line. This isn't something that the eye is doing, this is something that the brain is doing. Now, depth cues, which are essentially distance cues, may come from one eye or both eyes. We tend to depend a lot on binocular depth cues, unless perhaps you're a pirate. Binocular disparity is when the image of the same object on your respective eyes differs, but this difference diminishes with greater distance. Okay, so this makes it harder to judge the size of objects or the distance between objects the further they are away from us. We combine the 2D retinal images that we get from each eye to construct a 3D image of the world. Information from both the left and right visual field will enter each eye. Okay, so we get information from the left and the right eye there. We get information from the left and right visual fields going into both the left and the right eye. However, you can see there that the image falling on each eye will differ very slightly. This information is passed from the optic nerve to the optic chiasm, where it is combined and then splits according to visual field. It is then sent up the chain to the relay stations. So we know that the left hemisphere processes the information from the right visual field, and vice versa. The right visual cortex will be processing information from the left visual field because that information transfers over, but each eye does actually receive information from both the left and right visual fields. Now, the brain receives fragments of information from millions of axons that form the optic nerves. It combines and organises this information so that we perceive a scene of objects having different form, colours and textures and residing at different locations in a 3D world. We perceive a scene that is stable even when our bodies or our eyes are moving. This is because the brain keeps track of our own movements and those of our eyes and compensates for the constantly changing patterns of neural firing that these movements cause. It is because of the way that our brain organises this information, as well as visual experience, that we are able to see visual perspective, superimposition, relative size, height in a visual plane, texture gradient, and that we experience motion parallax. So let us have a look at one example of a visual illusion so that we can understand that what we perceive is not always based on reality. Now, edge detection is important for discriminating objects. The visual system searches for edges and enhances them so that we can clearly see them. This visual illusion is referred to as Mach bands. Lateral inhibition can explain why we see this illusion. The idea is that the colour across each band is uniform. However, we tend to see the bands as being darker towards the right edge of the band, whereas that the left of each band appears lighter. Now this is because the next band to the left is darker. And we go through a process of edge enhancement. So in the Mach bands illusion, the enhanced edges are illusory. So actually, even though this, for instance, this side of the band appears lighter than this side, it's actually uniform across. Now, Gestalt psychology argues that when it comes to visual perception, the whole is more than the sum of its parts. That is, processes of perception are designed to see the scene rather than bits of light here and there. So the perception that emerges from a physical scene may not be directly predicted by the sensory components that it is composed of, but emerges when we integrate the components into a whole. Figure ground organisation refers to the experience viewers have as to which part of an image is in the foreground and which part is in the background of a particular scene. In essence, we divide the world into two elements, the figure, that is the object of regard, and the rest, which is referred to as ground or background. And in many cases, we can choose what will be the figure. Now this is one example. You may see two faces looking at each other, or you may see a vase, but you can only see one or the other at any one time. You cannot see both at the same time. This is a version of the Rubin vase, which is based on the work of the Swiss psychologist Edgar Rubin in the 1920s. This is the Müller-Lyer illusion. Now, when asked which line is the longest, most people think the left line is the longer of the two. The fact is, they are both the same length. One of the explanations as to why we are susceptible to this illusion is because of our experiences of angled buildings. People who are from carpentered worlds, that is, worlds in which buildings are built from long, straight pieces of material that normally join each other at right angles, are more likely to be subject to the Müller-Lyer illusion. People from non-carpentered cultures that lack rectangular corners are less likely to be susceptible to this illusion, and therefore environmental experience influences perceptual experiences. Hello. Let us now turn our attention to the brain mechanisms of visual perception. What we have realised by now is that although the eyes contain the photoreceptors that detect areas of different brightness and colours, perception takes place in the brain, and it goes through a hierarchy of processing. Primary visual cortex receives most of its information from the visual relay nuclei of the thalamus. Secondary visual cortex receives most of their input from the primary visual cortex, and visual association cortex receives input from areas of secondary visual cortex. The primary visual cortex is located in the posterior region of the occipital lobes. Most areas of secondary visual cortex are located in two general regions, in the prestriate cortex and in the inferotemporal cortex. The largest area of association cortex that receives visual input is the posterior parietal cortex. And we can see these dorsal and visual streams here. So this is the dorsal stream, this is the ventral visual stream. And we'll come back to those in a moment. Circuits of neurons analyse particular aspects of visual information and send results of their analysis to another circuit which performs further analysis. Okay, the visual scene is broken down into modules and information is analysed in terms of orientation, thickness of lines, colour, movement, etc. And this moves up the stream, as it were. At each step, successively more complex features are analysed, and eventually the process leads to the perception of the scene with all the objects in it. The higher levels will interact with memories, and the viewer recognises familiar objects and learns the appearance of new familiar ones. Patients with damage to regions of the visual system have been used to understand the organisation of the visual system. Two visual streams have been proposed, a dorsal stream and a ventral stream. The dorsal stream flows from the primary visual cortex to the dorsal prestriate cortex and then to the posterior parietal cortex. The ventral stream flows from the primary visual cortex to the ventral prestriate cortex to the inferotemporal cortex. These two different streams of visual perception are thought to mediate different types of vision, which are outlined here. The dorsal stream specialises in visual spatial perception and in visually guided behaviour, whereas the ventral stream specialises in visual pattern recognition and in conscious visual perception. When the brain is damaged and visual perception is impaired, the patient is said to exhibit a perceptual disorder. Here are some examples of perceptual disorders. In all cases, there is no underlying impairment in sensation. The patient is able to tell light from dark and so on, and this is different to visual impairments caused by diseases such as glaucoma. So a scotoma is caused by damage to an area of the primary visual cortex. It is an area of blindness in the visual field. Deficits in the visual field can be detected using a perimetry test, which is also called a visual field test. Now, patients with a scotoma may not be completely consciously aware of the deficits. Okay, so this might be the result of completion if we remember what happens in the case of the blind spot. So, for instance, some patients, they have a deterioration within their visual fields and aren't aware of it because it's happened perhaps over a period of time and the brain has gone through a process of completion. Blindsight is something that we've come across before in our previous seminar sessions. Blindsight is caused by damage to the primary visual cortex. So, as we said in the previous slide, when the primary visual cortex is damaged, a person becomes blind in some portion of the visual field. Some individuals, however, can lose substantial areas of the primary visual cortex and yet show evidence of perceiving objects despite being cortically blind. This phenomenon is called blindsight. So blindsight is the ability of patients to respond to visual stimuli in their scotomas without conscious awareness of the stimuli. Although blindsight patients are unable to see properties of the object, they are aware of other aspects such as movement of the objects. If we take the case of patient DB, patient DB had undergone surgery for a brain tumour which required removing an area of the right occipital lobe. The surgery resulted in a scotoma, which is an area of complete blindness in the visual field. Patient DB could reach for moving stimuli, distinguish the orientation of a vertical line from a horizontal or diagonal one. However, when asked, he said he couldn't see anything when test stimuli were presented. Okay, so he lacked conscious awareness of the stimuli. So why can patients like DB make perceptual decisions despite being unaware of visual stimuli? Well, two neurological interpretations to the data have been proposed. The first is that the striate cortex is not completely destroyed and the remaining residual striatal cortex, which often referred to as islands, are capable of mediating some visual abilities in the absence of conscious awareness. And the second is that those visual pathways that ascend from subcortical visual structures without passing through the primary visual cortex are capable of maintaining some visual abilities in the absence of cognitive awareness. Now, the evidence is not conclusive for either hypothesis. It is possible that both mechanisms contribute to this phenomenon. Agnosia, in a more general sense, is a failure of recognition that is not attributable to a sensory deficit or to verbal or intellectual impairment. Visual agnosia is a specific agnosia for visual stimuli, in other words, an inability to properly recognise objects from visual information. Now, visual agnosics can see visual stimuli, but they don't know what they are. Two forms of visual agnosia are acknowledged, apperceptive and associative agnosia, though the existence of specific types of agnosia is a controversial topic itself in perception and neuropsychology. Apperceptive agnosia is the inability to recognise objects. Apperceptive agnosics have a severe impairment in the ability to copy drawings. So if we have a look here, the first and third columns are shapes which a patient with apperceptive agnosia was asked to draw, and the other two columns are the results of the patient's attempts. So we can see that there is clearly a difficulty in recognising the objects and the ability to copy them. In severe cases, patients generally show damage to a range of occipital areas as well as neighbouring brain areas. In less severe cases, patients may be able to distinguish between simple shapes when shown them, for example, a square and a circle. In these cases, brain damage is found in the posterior regions of the right hemisphere, although locations may vary regarding occipital, parietal and temporal lobes. Associative agnosia is the inability to make meaningful associations to objects that are visually presented. Associative agnosics can copy accurately but are unable to identify their drawings. They may not be able to draw the objects from memory. So copying a drawing may be done in a step-by-step manner rather than drawing upon perception of what an object is. If we have a look at the example here, they are quite good at copying, but might not be able to say what it is. So although patients may in some cases even recognise an object at a high level of information recall, for example, it is a bird or it is a dog, all the characteristics of that perceptual category will not be recalled. For example, does it build a nest or is it dangerous? In patients with associative agnosia, damage is generally found in areas representing the border between the occipital and temporal lobes in both hemispheres, though damage may sometimes be limited to one hemisphere only. Now, prosopagnosia is a visual agnosia for faces, so there is a difficulty in recognising familiar faces. So people with prosopagnosia can usually recognise a face as a face, but have problems recognising who the face belongs to. Some patients cannot recognise famous faces, their mother's face, or even their own face in the mirror. Now, the specificity of this disorder, in other words, it only being concerned with faces, raises the possibility that faces have particular significance as visual stimuli. It also highlights that the ability to recognise a specific type of stimulus, for example, is it a face, is separate from the ability to recognise specific instances of a stimulus type. So, for example, is it John's face? Prosopagnosia is often associated with damage to the ventral stream at the boundary between the occipital and temporal lobes. This region, or part of this region, is referred to as the fusiform face area, or FFA, and parts of it are thought to be selectively responsive to human faces. Some evidence suggests that the patients may be able to unconsciously recognise familiar faces. Achinotopsia is a deficit in the ability to see movement progress in a normal, smooth fashion. It can be triggered by high doses of antidepressants, and it's associated with damage to the middle temporal area of the cortex, or also referred to as V5. Middle temporal area appears to be involved in the perception of motion. Some areas of primary visual cortex might respond to movement, but they do not provide information about the direction of movement. Now, here is the further reading for this lecture. The Martin Carlson Buskist chapters 5 and 6 are really the essential reading, but if you want to find out more about it, have a look at Pinnell and Ising and Keane. Here is a link at the bottom to some psychology experiments which provide you more of a hands-on experience with those perceptual illusions that we spoke about earlier on. There are also some helpful videos to watch to find out more about the perceptual disorders, which you can have a little look at to get a better grips with what they look like in practice. Okay, the end. Thank you for watching.